Welcome to Brain Ponderings, conversations with scientists at the forefront of brain research. I'm Mark Matson, the host. My guest today is Vilhelm Bohr, Will Bohr. He made pioneering contributions to understanding roles of DNA damage and repair in the nervous system, the brain in particular. And he's looked at this from a number of different angles in terms of normal aging and also Alzheimer's disease. And also there's these very interesting premature aging syndromes, the progerias, which he's worked on. And I'm going to talk with him about his work on kind of the molecular level things going on in the nervous system and all these conditions. So Will is former chief of the Laboratory of Molecular Gerontology at National Institute on Aging, the NIH. He's a professor at University of Copenhagen. And so Will, you grew up in my father was an attorney and my mother was a nurse. But you grew up in a family where I can kind of envision the home environment. There was a lot of science being talked about.
Sure, yes, there was, yeah. Mostly physics, which I got interested in for a while, decided was too difficult for me. Hello? Hello?
Yeah, so
yeah,
his grandfather was Niels Bohr, and his grandfather Age Bohr also got a Nobel Prize in physics. So he grew up in a physics background, and as you'll see, he went on to medical school. And so I kind of kept filling him in on, your lineage.
Yeah, sorry about this interruption.
Yeah.
But so yeah, I grew up in that environment, but I was decided to go into science and medicine, and I trained in medicine. Then I went into scientific work after that, and I went to work as a postdoc in Copenhagen, then went on to Stanford. And after that came to NIH, first to NCI, and then later on to NIA, where I have been until last year.
And I went on PubMed and, scrolled down to your first scientific publication in 1985. It was in Cell, the top journal. And that was when you're doing a postdoc at Stanford, and that's,
yeah.
And I don't know if you can hopefully you can remember that work, but you're looking, I guess, in cultured cells at DNA damage to specific regions of the genome and trying to understand whether DNA repair is better or worse in different regions. Is that?
Yes. But before I get into that, I just wanted to tell you that I've really enjoyed working with you, Mark, for many years. When we worked together at NIA, it was always very stimulating to discuss with you, and you've really been a very good guidance for me to get more into neuroscience. And I really, we had many postdocs between us, which was great. It was a good environment. We still have things going on, and we managed to co-lead some very interesting projects. So I really appreciate that time with you there. I wanted to say that. Yes, I started, that was not my first paper, but I had, I got early on, I got interested in DNA repair. I felt that goes all the way back to the 70s. I felt that was such an interesting thing, and that we were so exposed to damage to DNA, and it really presented a huge problem. And I have been interested in that ever since. I felt that was a very profound problem. And so I got to Stanford, and at that point, I got very interested in how individual genes were repaired. Usually in the work we did at the time, it was the whole genome. You analyze DNA repair in the whole genome. So it was a novel idea at the time to see if regions of the genome were repaired differentially, meaning that DNA repair wasn't just the same everywhere, but changed with different parameters. And we were very interested in chromatin structure. And the idea at the time was a lot of people thought about that DNA damage and its repair had a lot to do with accessibility, the openness of chromatin structure. And so I took a look at an active gene, compared it to an inactive region, and we found a big difference. And that difference was then called preferential DNA repair, and meaning that certain regions are repaired better in the genome than others. And that has been shown now in many circumstances. And then that work, we continue to work on something called transcription coupled repair, meaning that not only is an active gene repaired better than an inactive region, but also the transcribed DNA strand is repaired better than the non-transcribed strand. And that was something that a lot of people also then started working on. And a mechanism that still is discussed a lot in the field, but we have never found the total underlying mechanism of how that works. Whereas we found some mechanism of how this preferential gene repair works.
And so there's a number of ways and enzymes involved in mechanisms of DNA repair. Some of them are probably more important, or I guess definitely for dividing cells, because the repair kind of happens, in conjunction with the DNA replication but then neurons, like in our brains, right? They're around their whole lifetime and they don't divide. So can you kind of briefly, but it's very complicated, but try to make it as easy as you can. What are the different kinds of DNA repair, and what do you think is most important for nerve cells?
Yeah. So we were early on, a lot of DNA repair was done studying UV lesions, historically, the exposure to the sun. And those are more well-defined damages to DNA, because very unique form and a few forms of DNA damage there. And then later on, with all interest in the nervous system and other things, we got more interested in oxidative damage and a pathway there that's called base excision repair, which removes these oxidative lesions. So DNA repair has many systems. It has nucleotide excision repair, which is the UV-related form. And then it has base excision repair for oxidative damage. And that's what you and I mostly worked on, because it's related to oxidative stress in the brain. And then the double-stranded break pathways, which in themselves are a number of different systems. And that then later became a focus of interest, because these double-strand breaks in DNA introduced by damages, for example, X-rays and many other types of chemicals, they are very detrimental to cells. So they are thought to be more dangerous than oxidative lesions and UV-induced damage, for example. So I've been interested in all those systems, but in relation to how we think about the nervous system, as I mentioned, base excision repair, oxidative damage removal seems to be on the forefront. And that is also, I started working with UV in the early years of this work, and then later drifted into this oxidative damage, and the work that I've done in the nervous system, and then you and I have done, as mentioned, is more related to that pathway of DNA repair.
And so in our cell, So you were at the National Cancer Institute for a while, so I think many people know that ultraviolet light can increase risk for skin cancer, and that with aging, incidence of many cancers increases. Oxidative stress increases with aging. Maybe DNA repair ability is impaired with aging, and that can lead to increased mutations that can cause cancer. But in the preceding podcast, Will, I talked with Professor Zaretsky on telomeres and telomerase and kind of non-telomeric functions. But we talked about DNA damage at telomeres and how it can trigger either apoptosis, cell death, or senescence. But on the other hand, if you have a DNA damage, say in p53 gene that causes a mutation, then that can lead to cancer. So can you talk a little bit about this base excision repair and what are the different steps and enzymes? And then we get to talk more about it.
Yeah, sure. So the steps are very simple at base excision repair because it's really firstly an enzyme, a glycosylase that recognizes damage in the DNA, and then an AP endonuclease creates a backbone break in the DNA, and then you have a polymerase step where you repair the damaged strand based on the intact strand as a template. And then finally, that piece is ligated into DNA. So it is a fairly simple process compared to other DNA repair processes. And some of the steps here are more essential than others. There are many glycosylases, so many different enzymes that can recognize lesions in the pathway. There's only one AP endonuclease that creates that endonucleolytic cleavage. And then there's really one major polymerase that creates that step of recovering the strand and filling up. And then there's a number of different ligases. So some of the steps, if you knock out these steps in mouse models, then they are not viable.
Yeah.
Whereas in other steps, they are more redundant in this process.
And also, in a little bit, maybe we'll talk about this, but of course, most of the work on DNA damage and repair is focused on the nucleus and the gene, the DNA in the nucleus, but there's also DNA in mitochondria, and there's evidence now that this base excision repair can occur to that.
Yeah. And that's a really interesting question, and it's something that I've worked a lot with for many years, is this difference between the repair in the nucleus and in the mitochondria. And obviously, while to some extent mitochondria are dispensable, the mitochondrial genome as such is essential. So it's very important to have that repaired as well as it is for the nuclear DNA. And largely, a lot of people have been interested in it. It's difficult to do those studies because you have to be able to, in many cases, purify the mitochondrial DNA and extensive steps to really analyze the mitochondrial DNA. There was some early work from Bruce Ames that suggested that when you are exposed to damage, oxidative damage, for example, then you get more hits in the mitochondrial DNA than in the nuclear DNA. So there's been some thinking that maybe mitochondrial DNA is a sink for damage. But I think that is not really the case. The damage that occurs in mitochondrial DNA is at the same frequency as what happens in the nuclear DNA. But the DNA repair processes differ considerably. importantly, the nucleotide excision repair process, the one that we talked about before for UV, does not take place in mitochondria. They don't have that set of enzymology. They seem to have a fairly similar base excision repair process as does the nuclear system. They don't have clear pathways for double-strand break repair. They have a system for mismatch repair that is different from what is seen for nuclear damage repair. So the base excision system has a lot of similarity between mitochondrial DNA repair and nuclear DNA repair, although there are some enzymes that differ.
And so with mitochondria, so cells have many mitochondria, and you can imagine if DNA gets damaged a lot in one mitochondria and it's not repaired, the cells have this mechanism that you've studied to identify that mitochondria as being damaged and dysfunctional and get rid of it. And then mitochondria divide so they can be replaced. So in that sense, it's kind of a different way, a way that the cells can remove the mitochondria so that they don't cause problems, I guess.
Yes, to some extent, yes.
Yeah.
So to what extent, how, that's a very interesting question, I think. And to some extent, they can remove themselves, but to some extent, they need to be, they restore themselves. And when exactly that happens and how much that relates to the amount of damage that's introduced is a really interesting question, I think.
Yeah.
So that is still unclear. People, I think, tend to think about it as if you have a certain amount of damage, then you can degrade the mitochondria if it exceeds a certain level. But if it doesn't, then it will restore themselves, although, for example, UV is not repaired in mitochondria. So for some cases, they need to be degraded. I think it's a very interesting and open question as to what repair systems exist for the double-stranded break repair pathways in mitochondria. And largely, many of the pathways known in the nucleus do not exist in the mitochondria. There's a lot of debate as to whether there are some possible pathways mitochondria for this.
And you had this, and related to this, you had this work where you looked at the effects of DNA damage in the nucleus on the mitochondria, and we're kind of studying this signaling mechanism between the nucleus and mitochondria, or at least you had a review on it.
Yeah, no, I have been very interested in the signaling pathways, and there are many. And I've also looked at the relation between damaged DNA and the mitophagy process. So I think we know of many signaling systems between the two. P53 raw signaling and things of that sort I've looked at over time.
And the sirtuins seem to play some role. And
well, yeah, so sirtuins, some are nuclear, some are mitochondrial. For example, sirtuin 3 that both of us have worked a lot with is in mitochondria and seems to be playing an important role in many things. And you and I were quite, I mean, you were really a stimulator for me to work with sirtuin 3.
Yeah.
Because you connected it to many things. And so I have been interested in its function, and also it plays a role in a base excision repair in the mitochondria. It directly deacetylates OGG1, and we thought maybe other steps of mitochondrial base excision repair, which we never completely finished. But of course, SOD2 It's not directly a DNA repair enzyme, but so
it could reduce SOD2, the antioxidant enzyme. It should be able to decrease the amount of oxidative DNA damage.
yeah,
So I guess this SIRT3 seems to be, influencing multiple proteins that have roles in preventing or repairing damage in the mitochondria.
yeah, it seems to be very important in that regard. And it also now there's sirtuin 7 and others that are targeted mitochondria that are less clear what they actually do, but could also play important roles in this mitochondrial metabolism.
And is this what? So what led you? So some of these sirtuins are dependent on NAD,
yeah,
nicotinamide adenine dinucleotide, and so this might be a good point for you to talk, because you have a translational research program where you were, looking in various models, premature aging syndromes, Alzheimer's mouse model that
yes,
you developed with our help.
yes. And that was something you and I discussed a lot. So I've been interested in premature aging syndromes ever since I started at the NIA, and even before that, in 1992, we were interested in why these diseases had such a close association to aging. They were accelerated aging. And a number of these diseases have DNA repair deficiencies. They are monogenetic. So I felt that studying those diseases would give us a good insight into, by extrapolation, to the aging process, and also connecting to the DNA damage process and DNA repair. So it was a very exciting area. First, one of the classic diseases that people have been interested in for many years in aging is Werner syndrome, which is a helicase involved in DNA repair that's mutated. And I've seen some of those patients, and I've worked a lot with this and visiting their clinics in Japan, and they are strikingly aging-like. So from early on, they develop so many features of the normal aging process. They live to maybe 46 years old. And so it is striking. And of course, it's different from normal aging, but nevertheless, it resembles normal aging. We did some early work looking at protein expression patterns and found very considerable similarities between Werner's and normal aging. So in the field, it goes back to George Martin, who was a leader in the field of aging, you remember, and he stimulated me to work with Werner and gave very inspired lectures about how we should understand the mechanism underlying these diseases. So I started working with Werner at the more biochemical level. We spent a lot of time purifying the protein back in the early 90s, and were able to look at protein interactions as a means to find pathways in which Werner participated that would then explain the aging pathways, if you will. So that's one disease I've been quite interested in.
I should say, Will, well, it's on my mind. So you mentioned George Martin, the 3xTGAD mouse model that we used a lot, and then
I use a lot.
You use a lot. So the way that was developed was very interesting. So when I was at University of Kentucky, we were doing a lot of work on amyloid beta peptide, and had this evidence it induces membrane-associated oxidative stress and can cause a lot of problems in neurons and that. But then, and then that was at the time that the first genes that were linked to Alzheimer's disease were identified. initially the amyloid precursor protein gene, but then presenilin 1, which there are many more families, particularly down in South America, where they have this dominantly inherited disease. And this presenilin 1, its function, or one of its functions, is to cleave the amyloid precursor protein and liberate the amyloid peptide that then aggregates and causes problems. And so anyway, I guess I saw George at a meeting or one of his postdocs or something, and then you know, we developed, it's actually him who actually made the mice, but then we characterized and made presenilin 1 mutant knock-in mice.
Mm.
So instead of just putting the gene in and overexpressing the mutant protein at very high abnormal levels, we put a mutated region of the human gene into the endogenous mouse presenilin 1 gene locus. So its expression is controlled by all the same, transcription factors as presenilin. So these mice, we let them age, they develop zero amyloid pathology. And it turns out the reason is that the amino acid sequence of the amyloid beta peptide in mouse is different than in humans. And the mouse amyloid beta peptide doesn't aggregate and toxic. But we did find some abnormalities in these mice in terms of calcium regulation and vulnerability of neurons. But then the way the 3xTGAD mice were made was we sent some of our presenilin 1 mutant knock-in mice to Frank LaFurla at UC Irvine. And what he did is very cool. He bred the presenilin 1 mutant mice, and then he took out from the uterus the one cell stage embryo, and then he injected that cell with genes for mutant amyloid precursor protein and mutant tau, this protein that accumulates. And so that mouse had 3 mutations. So it was kind of a really cool way of making a multi-gene transgenic without having to do all this interbreeding and worry about the genetic background. But anyway, those mice now are widely used.
Very widely used, yes.
And they develop age-related amyloid and tau pathology. So
yes.
then you came to me and said, let's collaborate. And so tell everybody what you did. You crossbred those with DNA repair deficient mice.
Well, now I didn't go from my the connection between what I wanted to say about these rare diseases of accelerated aging and this mouse model here, but it's fine. We can go this.
Yeah, let's go. We'll come back to it.
But we'll kind of come back to it because I guess yes, we maybe come back to either one, but we could to answer your question about this mouse. Yes, I came to you because given this development that I'll then get back to of why we really got into Alzheimer's, that was a progression of ideas from using these rare diseases, plus that I, in fact, worked as a neurologist in Copenhagen for some years before I went into to more spending time in the scientific side. So I had a real interest and experience in neurology, and I wanted to get back to that. And so when we had, based on a combination of things, including that I always felt that DNA repair was important, and then at the time that we began to work, it was that base excision repair process that I was interested in, of oxidative damage removal. And so I want to ask the question of how that process really played into Alzheimer's. And that was where you advised to use that 3xTG mouse, which we then chose for this work. And then we were able to, based on some other we also, with your collaboration, had a very interesting collaboration with the Kentucky Brain Bank
Yeah.
and Macclesbury and that group there that you knew, where you came from there. And we were then, we started looking for the DNA repair capacity of basic system repair in postmortem brain.
Yeah.
And that was really goes back to 2007. We published this paper, and you were involved advising there with and using the postmortem brain. And it was in those studies that we found that there was indeed a basic system repair deficiency in human postmortem brain. And it was in AD from human postmortem brain. And it was more progressed when you went from MCI to AD. So there was an intermediate step of MCI, mild cognitive impairment, and then to AD, that the DNA repair got less efficient. And we were able in those studies to target it to that step that I mentioned before called the polymerase step. That is the Pol beta enzyme that is a critical enzyme in basic system repair. And when we analyzed this, we could see that the amount and the activity of Pol beta declined with the loss of cognition. And we did activity studies. So we grinded up the brains and we had various controls, and we looked at the actual DNA repair activity by throwing in some substrates for these enzymes so we can measure their activity. We later did that in mitochondria too from those brains, and we're able to also see that in mitochondrial DNA repair, that was also the polymerase step that was deficient. And interestingly, the decline in the activity of Pol beta correlated inversely with the Braak staging of those patients. Do you remember that? So we actually had a very nice kind of finding that it was a targeted deficiency in Pol beta in the brains declining with declining cognition. So in terms of trying to limit using the mouse model of 3-XTG to try to study this phenomena, we then were able to make a heterozygote deficient Pol beta mouse in the 3-XTG.
Yeah.
So you can't completely remove Pol beta from this mouse because then it doesn't survive at all. As I mentioned, these enzymes are essential. So we did 50%, and with 50%, we could then see that we got a considerable worsening of the progression of AD. We could see that we had we got sort of a humanized AD mouse model. We had neuronal death. We had declining hippocampal volume. We had a lot of interesting deficiencies. We had an olfactory deficiency, something I've been interested in lately. So we really could show in this mouse model that when we were able to, when we made it deficient in this important repair capacity, then we got much worsening Alzheimer's features. So it suggested that there was an effect of DNA repair in preventing the more severe Alzheimer's.
Okay, and then do you want to talk about the NAD? Like
I will. So what the NAD had to do was that, again, in these many premature aging disorders that I've studied, we noted, because again, I've been interested in the neuroscience element of it, so we noted that some of them had very severe neurodegeneration. So there's some of these premature aging diseases, and Cockayne syndrome is one of those. And I've been interested in that disease for a long time, also because that disease has a particular deficiency in this transcription coupled repair that we talked about earlier. But anyhow, it is a DNA repair deficiency. And we had a seroderma, which is a well-known disease also with premature aging and premature cancer, in fact. And then we had ataxia telangiectasia, which is a very severe neurodegeneration in the patients as well. And we were kind of wondering why these diseases had so much neurodegeneration, and we were thinking in terms of mitochondrial dysfunction that could lead to that. So we did many, many papers on that in that period of time, analyzing this in different ways, using mouse models, using cellular systems, using human cells, using tissues from humans, and then getting increasingly into the nematode systems, which again, you were a big help in, because Mike there, he was, you had that set up there from the days of Kathy's lab. So we were really benefited from using that, and the nematode is a wonderful system to work with.
Yeah.
And I didn't get support to keep that going, but I would have really felt it was very important for the Aging Institute to have that.
Do you know if they still, have they kept that? Did Rafa keep that or not?
No, they didn't keep that, because they just, and that's really too bad in my opinion, really.
Yeah.
So we were just, hello?
Yeah, so the value of the worms, the C. elegans, the roundworms, that their genome's been completely sequenced, and there are mutants of any gene in the whole genome that there's a place you can just essentially order them from. So you can order worms that are deficient in a DNA repair enzyme, or whatever, mitochondrial protein, and so on. And a lot of these genes from worms to humans are conserved. And also in the worms, there were Nobel Prize given, actually more than one, I think, for worm work. So the complete development of the whole worm, which has like 1,000 some odd cells in its body, is known. There are, I think, maybe 300 some neurons. Yeah. Everyone's known. You can knock out, or I guess overexpress, any gene in only certain neurons. Kathy Wolkow did some of that work in my lab.
Yeah.
Other genes. So
yeah, sorry for my little interruption, but I wanted to say that one of the things that fascinates me so much about the nematode is that you can also study behavior there. Not just lifespan, but actually memory. So they have this chemotaxic feature that you can use for memory, and the AD worms have loss of memory, and we can screen these worms for things that stimulate the memory. I'll get to that. But
the chemotaxis, that's essentially like olfaction in humans, right? It's
it is a mixture of things, but it depends on what you use for stimulation. Then it can be more sensory, more smelling, and other things can be more like for memory. But I wanted to get back to this development. So we were looking at these findings, asking why these premature aging disorders had such severe neurodegeneration, and through using all these model systems collectively, we found that they all had severe mitochondrial dysfunction, and we could look at various mitochondrial functions and conclude that it was mainly the process of mitophagy that was deficient. So meaning that they were not able to get rid of the damaged mitochondria. So mitophagy is the process by which the mitochondria merge with lysosomes to get rid of the damaged mitochondria. And that targeting mechanism is essential. And if you can't get rid of damaged mitochondria, they accumulate, and that can be a major underlying cause of neurodegeneration. So in the process of that, we looked at many mitochondrial parameters and metabolites, and we found that all throughout we had a much lower level of NAD in these conditions. And we then explained that by a DNA repair process and a DNA damage process, because when you recognize damage, there's a process called pyrylation, by which PARP1 post-translationally modifies DNA and other proteins. And then we could show that that process, we knew that the process would use NAD as a substrate. PARP1 uses NAD as a substrate. So we could show in our systems with all of these conditions that there was a very highly increased level of pyrylation called hyperpyrylation, simply a very, because of the damage accumulates in DNA, you get a lot of this pyrylation. And we then at that point speculated that that was the way that NAD was removed from the cell. And at the time we first saw that, people would question, is it possible that you get so severe deficiency in NAD by this hyperpyrylation process? And now I think it's more widely accepted that that could be the explanation. Yeah. So then the interesting thing is that this idea was that when you have a lot of damage in the genome, you deplete your NAD pools by that process. And we then went on to show that it wasn't only in those diseases, but also a number of the other premature aging diseases where damage accumulates, like Werner syndrome, like aprotaxin. And then we further found that in many cases of Alzheimer's, where damage is well known to accumulate in the genome. So what we had seen very broadly is this connection that when you have a lot of damage in the genome, you seem to deplete your NAD pools.
And that fit with data from other, measures and indices of the ability of neurons to get enough energy, in aging and Alzheimer's. Impaired mitochondrial function, so less energy. And then we did all this work on excitotoxicity.
Yes.
Actually, that's what my book is a lot about. But anyway, neurons they're active all the time, right? And that's a stress on them. And this is something we'll talk about in a minute, I think. So most of the neurons in the brain are excitatory. They use glutamate as a neurotransmitter. It's an excitatory amino acid. So when neurons are active, that's a stress on them. There's depolarization of the membrane, sodium influx, calcium influx, free radical production, increased mitochondrial. It makes sense. So when the neuron's active, they need more ATP. So the mitochondria increase their electron transport to produce more ATP, but they're also producing more free radicals. So we get more DNA damage. And this is something, so why don't you talk about this? This is one of the very interesting findings we had, that kind of normal levels of activation of glutamate synapses increases DNA damage, but then it's repaired.
Yeah, no, I like that study a lot that we did together. That was this Jane Lin study, and that study suggested, and we both talked about this, that when we have low levels of glutamate, micromolar, a few micromolar or something, then we elicit oxidative signaling process with ROS formation, and that ROS formation actually, through signaling, activates I think it was the CAM kinase
Yeah.
process, right? And then so we actually have a loop whereby that damage activates DNA repair enzymes. And you and I reported that was AP endonuclease in Cenk Lin's work.
Yeah.
And later on, we and others found that it was also Paul beta, actually. So the funny thing is that when you just think
Yeah.
or just anything you do with your neurons, you elicit a small amount of glutamate signaling that causes oxidative stress and causes DNA repair processes. And so DNA repair occurs just under normal thinking. That's what we concluded there. And with physiological levels of glutamate. And I mean, we had many plans at the time to do more things. We put these mice in a spinning wheel. We did exercise things and looked more at it. And I think that was such an interesting aspect of our collaboration.
Yeah, you know what, we'd have, periodically I'd see you or someone from your lab give a talk, to the group there at NIA, and, that all the work you did on kind of the nuts and bolts of how DNA repair works and so on. But multiple times I'd ask you, well, how can we enhance our DNA repair, right? And so we figured out a few experiments. One was that, but the other was the exercise, because there's a lot of evidence that physical exercise, like you play soccer, or, I had a running group, it's good for your brain. And yeah, so Jenkins did find, right, that if you compared to mice that are sedentary, if you have mice that are in running wheels, they kind of beefs up their DNA repair ability.
Exactly, yeah, Paul beta and AP endonuclease, yes. Maybe I'll just finish the NAD thing a little bit, because what I wanted to say that was that we have then gone into a lot of translational studies using this NAD supplementation. And we found actually NAD itself can stimulate DNA repair. And there are early studies in this, and I've done a number of things with this, but also it stimulates mitophagy. So it has two targets that are very useful in the context of this. So we did a lot of treatment of AD mouse models, and you're involved in a lot of that work with NR, and found some extremely beneficial effects on the 3-XTG, also 2-XTG. And you remember a lot of this, and then we have done in recent years some interesting behavioral intervention where we used hearing in mice, and also found a very beneficial effect of NR in various circumstances. But
can you say what NR is and why you're using that?
Oh, yeah, sorry, yeah, sure. So NAD is synthesized in a number of different pathways, and one utilizes NMN and other substrates, and then nicotinamide riboside is a step towards the synthesis of NAD. And so there are many ways you can stimulate NAD synthesis, but some ways are more efficient than others, and some are more direct than others. There can be early degradation of some of the supplements. So what we have used consistently and with successfully, I would say, is nicotinamide riboside, and that is fairly similar to the effects we would see with NMN, nicotinamide mononucleotide. But nicotinamide riboside has then been sort of a useful tool to try to prove some of the stuff here that I'm mentioning, in the sense that we up the NAD levels when we give the mice this nicotinamide riboside. And it's in the whole system, but importantly in the brain. We showed early on in work where you were also collaborating with us, that when we give these mice nicotinamide, the 18 mice nicotinamide riboside in the drinking water, we also see an increase in the brain of NAD. So it either crosses the blood-brain barrier directly, or it does stimulate something that does. And so the effect is that we can up the NAD levels in the brain significantly with such treatment. And then we were able to show that we could greatly improve the AD to normalize AD in terms of cognitive impairment and other things. And one of the assays that we used was also in collaboration with you, which was LTP, long-term potentiation, which is a great way of looking at memory, if you will, or more importantly, maybe call it synaptic transmission, right? And so this was something that we did together, and that one of the most we've looked at so many features of these 80 mice, and the one that one of the most amazing changes is with LTP, where we completely normalize deficiency in AD. And so it has to do with synaptic transmission in the hippocampus. And we have in our studies with NR, also in the inner ear, for example, we return to that it really stimulates synaptic transmission.
And with that, did you just look at hearing loss during normal aging, or what was the?
Yeah, no, with that we've looked at hearing loss in Cockayne syndrome and in normal aging, but we've also looked at the cochlea and looked at hair cells and synapses in the cochlea, and seeing that pathways of synaptic transmission and other proteins involved in synaptic transmission are directly stimulated. So there's also other work in general that supports this, that I would say strikingly in many things. With this NAD supplementation, you have a very beneficial effect on synaptic transmission.
Yeah. And
other people, and also we have seen a big, very positive effect on neuroinflammation. So those are two of the things that dramatically improve with this intervention.
And you also, you mentioned ATM, ataxia telangiectasia.
Yeah.
Hard word to pronounce. But so that's the ATM, it's not directly a DNA repair. It's a kinase, right? It's a protein that phosphorylates and affects function of other proteins.
Yeah, it has many functions, and it's mainly a kinase, but it kinases a lot of the key players in DNA double-strand break repair.
I see.
So it's called as a master molecule of DNA double-strand break repair.
And for some reason in that disorder, so it's inherited, and so these kids, it mainly for some reason affects neurons in the cerebellum that control body movements.
Yeah, so it's in that family of these cerebellar neurons, and it's a Purkinje cell thing.
Yeah.
And then it also they have a very interesting severe immune defect, and the patients actually die from lung diseases very commonly. So it's a very interesting pattern, but it is clearly thought to be a Purkinje cell defect primarily. Now here is the most then translationally interesting part of all this is that we published this work that in 18 mice we could have a major improvement with NR of these functions. And now there have been 2 clinical studies of patients with AT where they've been given this NR, nicotinamide riboside, and there's substantial improvements.
Oh,
So I'm
well,
very excited about that this work has led to something where we see this helps the patients. And both of the studies, the last study was just published here with some collaborators from Norway who did all the work there. And the earlier study was in the Netherlands. And these are difficult studies. They're very, patients that are very affected. But it was, and I think that we now know that the dose was way too low because NR is safe and people can take up to 3 grams per day, and they only use 250 milligrams in those patients. But the results are pretty neat that there are some improvements. There are some neurological improvements in the latest study, rapid eye movements and other things that they measured. So I'm eager to help get a bigger study with higher doses through.
Yeah.
We also have seen in a clinical study from Werner's patients, that's not finished completely yet, but it looks somewhat hopeful that there is a little benefit there.
And in the Werner kids, did you use the higher dose or still the lower dose?
Still lower dose,
Okay.
because when in all these plannings, we were very concerned about how much to give these patients when they're so
Yeah.
debilitated. I mean, Werner's, the ATs are mostly children. Werner's are adults, but so I think that since we started those studies, the knowledge from the rest of the studies with this is we could go a lot higher in dose.
Now this is, people are taking this kind of random people that are health conscious, have heard about this kind of thing, and they're buying and taking NR. Are you, and since it's not harmful, I guess can't do any harm. I'm just wondering whether it's worth taking, like it's kind of a hedge against age-related problems.
Yeah, I mean, I would say so. I have worked with this for many years, and I've become convinced it's safe. I mean, I don't know that we really know what happens to normal individuals over a long period of time with this, because studies have been done for 1 year, and many of the results on, should we say, normal individuals are not that there are no changes. But my underlying idea with all this is that we need to target those individuals who have lower levels of NAD, and they could benefit from it. But it's not so simple to determine that, and we don't know where to look for the lower NAD levels in particular. I mean, it's not enough to look in blood, and we can't really do it very much in brain, although maybe a little bit. But that it's like so many other things, it should be individually targeted. So then some people are likely to get more of a benefit. Now, since there's no harm in it, I think it's also maybe not, but it's pretty expensive to eat every day.
Yeah. So we did work with this ketone ester.
Yeah.
And that's in clinical trials now, including Dimitros is involved in one trial, I think.
Yeah.
But it's in just kind of a general sense, it's a similar idea that, kind of boosting the energy availability. And with aging and really prominently in mild cognitive impairment and Alzheimer's, neurons have trouble taking up glucose, but they still seem to be able to take up the ketones
Yeah.
So the idea, it's a very rational thing, and the preclinical data are strong, whether it's with, NR or ketones. And also this relates to fasting and exercise.
Yes. And we did it. You and I did a study with this, and we have treated 3-XTG and 3-XTG Paul beta with this, both combined and separately. And we never, the postdoc left, and we never finished the whole thing with that. But there is some data that the Beach Laboratory people that are still there are trying to examine. And so there may be something we can get out of that. But that was this idea. You were so strongly in favor of doing this with the ketone. And I was at that time a little less, but I was convincible. So I was happy to join this effort. But I think the problem then was that given all the circumstances, it became such a huge project.
Well, and then, I guess that was before COVID hit, but when COVID hit, that pretty much shut down all research, unless you're doing research on COVID, right?
Yeah, that too.
Yeah.
That too.
Let's see. What else? So now you are you doing research in Copenhagen? Or
yeah, so I do a little research. I do a small group in Copenhagen, but I'm still here in Maryland.
Yeah. And you have any plans for writing a book?
No, I mean, I have thought about it. I'm involved in a number of papers and reviewing a lot of things, that kind of stuff. But I've always admired your writing skills. You're so quick and fast to write papers and books, and very efficient at it. For me, it takes a long time, so I haven't really considered that so much. But I mean, it's very worthwhile to do. But it's not a quick thing for me to write.
No, I mean, it's not real quick. It takes me probably a year to.
Yeah.
So what I do is just like 2 or 3 hours a day. In the morning when my brain is really sharp.
Yeah.
I just, and then the rest of the day or evening, I'll often think about what I'm going to write next, or what I've written, what I'm going to write next, and just kind of keep that going and chip away at it. But I mean, you've got some interesting things to talk about.
Yeah, I mean, I've thought about writing a book, or certainly thought about writing a lengthy review, but it's definitely on the agenda.
Yeah.
But maybe you can inspire me a little bit to do that.
Okay.
Kind of be fun to work with someone. I mean, I like to work with other people in these things. It's hard for me to just sit down and write by myself, but I can.
Yeah. Huh. Okay. Well, I'm working on a third book now on hormesis, adaptive stress response.
Yeah, you've always been interested in that.
Yeah. And this one's it's taking a long time because there's so much to talk about, and it's hard to figure out, what might be most interesting to the readers. So, yeah. Okay, let's see. Now the progerias, you mentioned a trial in Werner. There's so few of these people, right? I mean, they're very rare disorders. And but from the standpoint of age, I mean, so the group, who's the group over there? I don't know if it's in Netherlands or wherever. They were looking at well, yeah, they did calorie restriction and DNA. So what's been done with, we know calorie restriction, intermittent fasting extends longevity in lab animals, and then there's been some work done asking since these whether it's reduction in polymerase beta or
Yeah, so
Turner.
there has been a lot of work, and we did a study that I also think you and I collaborated on, where we looked at these mice under caloric restriction, and we looked at what happened to DNA repair, again, base excision repair. And we had that interesting finding that when you went on caloric restriction, the mitochondrial DNA repair was more stimulated than the nuclear DNA repair. It was sort of a targeted thing, the mitochondrial DNA repair increased. And but since I kind of just have come off of caloric restriction, because I feel that it's just, I would be more interested in intermittent fasting and some of the other things that you've also worked with, because I think it's just real caloric restriction is not a very good, useful intervention in humans.
Yeah.
So we would like to do something that has relation to humans. But there's a lot of old work on caloric restriction and DNA repair, but that finding is kind of interesting, that maybe it stimulates the mitochondrial DNA repair more than the nuclear DNA repair.
Now, obesity and type 2 diabetes are bad for pretty much every organ system, but they're not good for the brain. Has this been looked at, whether obesity, diabetes impairs repair? Is that known in the brain?
I think there are very strong connections to DNA repair and diabetes. There are direct pathway connections to DNA repair genes and diabetic pathways. I don't know about what kind of obesity studies might have been done, but of course we and others have looked over time at high-fat diet kind of situations, but I don't think that I can just readily think about some very clear connections that come to mind here with DNA repair. But it's been a while since I've thought about that. But I think I'm sure that high-fat diet relates to oxidative stress, of course, and so there's definitely connection again to DNA damage. And let me say that I've been working with DNA damage and repair for 35 years in the aging field here, and it's interesting how this kind of goes into focus and out of focus. And so there have always been people who strongly felt that the DNA damage was one of the most critical factors, if not, one of the critical factors. And of course, it's also the genomic instability is one of the major hallmarks of aging. But then people have strongly connected to that, in the field, and then it's gone back and forth a little. And I think lately, again, there's a lot of interest in that, but there have always been some divided opinions about this. And of course, some of us who have followed it very closely and spent so much time working with it, we would argue strongly that DNA damage is a really important element in all this, and others would maybe argue less. But certainly DNA damage connects directly to senescence.
Yeah.
It's the major cause of senescence. So I think, that is something people could agree would be a factor in aging. It connects directly to mitochondrial dysfunction via pathways that I've mentioned and others. And so it also connects to telomeric processing. You mentioned that a little earlier. And the DNA damage in the telomere has a lot to do with telomeric shortening processes. So I think that it would be, one can argue strongly that it is a very important aspect. And then we also have to think about the complexity of DNA damage. There are so many kinds of damage that we're exposed to, and there are so many DNA damages that we still don't know, that we need to find out more about. So there's a lot of, I would say, future potential in further understanding this and further understanding which one of these, all these many types of damage most closely relates to aging features we're interested in and things that of that sort.
Are there, so as far as mutations, you can inherit a gene mutation. Well, you inherit it one way or the other, but you can
Yeah.
have, for example, you can have parents who neither one has a mutation in a gene, but you could have a mutation because of
Yeah,
mutation occurring in the egg
yeah,
or the sperm, right?
yeah,
Or
yeah.
maybe even the early embryo. Um, is
So it's like,
there much work going on? There must be work going on DNA damage and repair
yeah,
and these.
absolutely. And so you have these more somatic mutations that may be more related to the things we're talking about. But there's a lot going on with that. And of course, we also would be interested in where these mutations that are induced mutations mainly targeted. And so, for example, one way of study is which genes get mutated, where we know that, for example, P53 is one of those target sites for these mutations. And so that's another interesting thing. And again, for example, we have, yeah, that is a really interesting thing. We know from earlier studies that when you look at humans, there is and you try to do DNA repair in humans, in human cells, that you can get roughly 0.8% decline with age in humans. So we have this number that suggests that we lose most of a percent of DNA repair with aging. And when people
That's per year, loss per year?
per year,
Yeah, per year. Okay.
And if you look at mutation rates and somatic mutations measured in genes that are targeted on a plasmid or something, then you get about that same number. So there is a sort of correlation between damage mutation and in aging here, where you can estimate maybe how much we lose of these capacities over time, roughly.
Well, that's so that's suggesting, you and me, we're getting maybe down a 50% reduction in
Yeah,
repair.
probably somewhat.
You need to exercise and take some NR maybe.
Exactly. And I think that you've been a big promoter of exercise, and I think there's absolutely no question now that exercise is critical, and also for the brain, right?
Yep.
And I guess the question that I want to ask you maybe is, how much do we have to do? Because you see, these people saying now you're supposed to do very active thing. You have to get your pulse very high, and others say you don't. And there's a lot of diversity. I mean, going for a walk every day is obviously really good, right? But do you have to?
Yeah, walking for your cardiovascular system, my opinion is you need a little more vigorous exercise, unless you're, really walking fast, walking uphill. Now I had a mountain bike accident a number of years ago. I had these surgeries. I couldn't exercise for a while. Then I got walk, but until I got back, being able this past year essentially to do vigorous activity on the mountain bike. So anyway, when I ran, I used to have resting heart rate like in the 50s, blood pressure 110/70 or even lower. And then during that time, I couldn't exercise. My resting heart rate went up in the 70s. My blood pressure was up something like 140/90. And then when I got back exercising vigorously, went down. So but as far as, I think walking is really good for your brain. I do a lot of good, and exercising in general do a lot of good thinking while you're exercising.
But I was talking to some experts, I was in Germany, that claim that you need to do very active things for a short period of time, and then that's what you need to do. And then I've read other literature, a lot of that says that you just need to do a little. What
It depends. I mean, the question is how much it's too much, I guess. But yeah, some exercise is better than none, and then there must be, and it could vary somewhat between persons and their genetics. You know, are you more or less prone to whatever cardiovascular disease and so on? Yeah, so I don't know. That's the same thing with, you know, the intermittent fasting story. I think, you know, these short, you know, daily fasts of 18 hours or whatever, that's no big deal for us from an evolutionary perspective, right? So I think that's kind of a very minor but beneficial, you know, intermittent or kind of a daily stressor, mild stressor.
So you recommend 18 hours of fasting per day?
Yeah, that's what a lot of the clinical trials are doing now.
Yeah.
And that's what I do. I just skip breakfast and then I exercise right before I eat lunch. I eat all my food between noon and 6 p.m.
Yeah.
And, if you normally eat breakfast and then you start skipping breakfast, you'll be hungry and irritable for, you know, number of days, maybe up to a couple weeks, and then, but then that will disappear, completely disappear after you've adapted to it. So yeah. Okay, well, I know you said you sent me a message saying you had to get out of there pretty soon. And it's really good to see you. We'll have to keep in touch, maybe get together in person sometime.
That would be really nice, Mark. I would enjoy that. Yeah.
Okay, well, have good holidays. It's good to talk with you.
Yeah, and thanks for having me. And I really like your show there. I've seen a couple of other discussions. You're doing a great job. I think it's a really important service that you do there. And best of luck to you going forward, Mark.
Okay, but well, think about writing a book.
Yes, sir.
Okay, I'll buy it. Bye, Will.
Bye.